Hello, welcome to our webinar, Testing for Coliforms and E. coli. My name is Clint Burkhardt. I will be the host for this session. It is now my pleasure to introduce our presenter for today, Shawn Dubois. Shawn is a senior scientist here at IDEXX. Shawn has a BA in biology and a BS in environmental science with a concentration in marine biology and marine ecology. Shawn has been part of our water business for 20 years, working in research and development, and now in his current role, he manages the product support group. So without any further ado, I'd like to turn it over to you, Shawn.
Great. Well, thank you, Clint, and thank you everybody for joining us. So just kind of talk about what we're going to go over today. We're going to go a little bit over what an indicator organism is and why it's important and how we test for them. Specifically looking at presence, absence, and quantification using enzymatic methods. Going to discuss Colilert, Coli18, Colisure, and the new product IDEXX Tecta for testing of coliforms and E. coli. And then finally going to touch on a little bit on quality control testing. So to start off, what is an indicator organism? So when we talk about testing for water quality, there are plenty of pathogenic viruses, bacteria, and other organisms that could be present in contaminated waters. There are way too many for us to be looking for, and they can be very difficult to find, dangerous, and very time-consuming. So rather than looking for all the possible pathogens that could be present in contaminated water, instead we look for an organism that will be present when waters are contaminated. Kind of looking for smoke instead of looking for the fire. So when we talk about indicator organisms, there are some kind of a checklist of what would make an ideal indicator organism. First, again, we want something that would be present when a pathogen is present. On the same side, on the opposite actually, we want them to be not present if the waters are not contaminated. We want them to be in very high numbers, especially higher numbers compared to pathogens, because we want to be able to find them easily in just even a small water sample. We want them to be better suited to survive in the water environment, so that way we have the opportunity to find them when we're testing our waters. And then above all, we want something that's easy and safe to analyze. We want to make sure that the analysts who are doing the testing are able to handle this without any concerns for safety. We want results quickly, so that way we can take action as fast as possible. Relatively inexpensive. And finally, we want a test that's accurate. We want to be able to say with confidence when we are testing our waters that we're getting the correct result. We want to say that if we're finding a positive, that we know that there isn't contamination and that we're going to mitigate the issue. And at the same time, if it's negative, we want to say with confidence that this water is safe for people to either drink if it's drinking water or to utilize if it's recreational waters. So when we often think about indicator organisms, the one that pops to mind the most is E. coli. E. coli is a major inhabitant of our intestinal flora. It is found in very high numbers, and therefore it is also shed in very high numbers in fecal material. And the really big important thing here is that it does not naturally occur in the environment. If we're finding E. coli, that means there has been some kind of contamination event that has occurred. So this makes it the perfect indicator organism for fecal contamination. A couple other things about it: it's a gram-negative bacteria, and it's also thermotolerant. And what this means is that it's able to survive in a wide range of temperatures, up into like 44.5 for most species of E. coli, strains of E. coli. So it's a great indicator organism for fecal contamination. Now, when we talk about E. coli, coliforms, or really kind of any indicator organism, one of the important things we want to understand is this growth curve. This is kind of what the life cycle of the bacteria will be when it's put into like kind of the perfect environment. So when we're talking about testing waters, we're testing for the indicator organism, and it's most likely been through some kind of treatment process. So if it's drinking water, you can have chlorination, ozonation, or UV. And what we're doing is we're basically trying to kill off the bacteria or injure it enough where it won't be as active. So what that means is when we're testing for it, we need to help it recover and become healthy again. So when we take that bacteria that's been injured and we put it into our testing media, we're giving it kind of the imperfect environment to grow. And what we want to focus here is these first two phases of this growth curve, because this is what's going to show us how we're recovering that bacteria. So the lag phase is the part where the bacteria is in place in this really nice environment, lots of nutrients, and what it's doing is it's repairing itself. So it's repairing any kind of holes that it has in its cell wall, and basically becoming really healthy in order to start growing and increasing in numbers. After this lag phase, where it's been recovering, the next phase is the log phase. And this log phase is actually when we're going to start having that population growth. So if we start off with 1 E. coli, that 1 E. coli will become 2, that 2 will become 4, then 8, 16, 32, 64, and basically just population keeps on doubling in size. And so this is really important because this is when we're going to start seeing that indication that that bacteria is present when we're testing our waters. So again, we really want to focus on the two parts, the lag phase, which is the recovering of the injured bacteria, and then the log phase where we're going to start seeing that growth. So when we talk about testing, there has been a change over time as to how we do test for those bacteria. Initially, the first kind of testing we had was multiple tube fermentation. It is a rather lengthy process. It takes up to 60 steps to run this kind of testing format, and it can take up to 4 days to get a confirmed result. So it's a very inefficient system in the fact that it takes a lot of steps. There's a lot of points where errors could occur, and it also takes up to 4 days to get a confirmed result. So it's not exactly quick. Also, it's not exactly the most accurate of tests either. Next, we have membrane filtration, which is a vast improvement over multiple tube fermentation in that the number of steps is reduced to about 20 steps total for the process, with confirmed results at 24 or 48 hours, depending on the method. So definitely an improvement over time in steps, but it's still a rather technique-based testing where there's a lot of points and where you could have some errors again. And then finally, we hit enzymatic tests. Enzymatic tests are more accurate and quicker for results and easier to run. So total, depending on whether or not you're running presence, absence, or quantification, it's about a total of 3 to 8 steps. And some examples here, we have Colilert, which is a 24-hour test, Colilert 18, which is 18 hours, Colisure, which is 24, and the new product IDEXX Tecta, which can have results as early as 2 hours, depending on the contamination of the water. So a lot easier, a lot faster, no confirmation steps required. So now I'm going to go a little bit more into the actual process of running enzymatic tests in either a presence-absence format or a quantification format. So presence-absence is pretty simple. It's basically a 3-step process where, in the case of Colilert, Colilert 18, and Colisure, you would take the reagent, add it to your 100 ml sample, mix well, incubate, and then after incubation, read the result. So it's pretty simple. It is basically a 1, 2, 3 step process. Basically kind of say if you can make Kool-Aid, then you can run these tests. Pretty straightforward. Quantification requires an extra couple steps and some additional equipment. Again, this is for Colilert, Colilert 18, and Colisure. So first you would need the Quanti-Trays, and the Quanti-Trays are the actual quantification device for the test. There's 2 different formats. There's a 51 well Quanti-Tray and a Quanti-Tray 2000, which has 97 wells. There's an associated Quanti-Tray rubber insert that the tray will go into, and then the actual Quanti-Tray sealer itself. So again, it's a very simple process to do quantification, a few more steps than presence-absence, but you still start off the same way where you take your sample, add your reagent to that sample, so Colilert, Colilert 18, or Colisure, mix to dissolve. That sample mixture will then get poured into the Quanti-Tray format of choice. That will then get put into that rubber insert, and then that insert and the tray will go into the Quanti-Tray sealer. And basically what's happening is through a combination of heat and pressure, that sample is being moved up and filling all the wells in the tray. And at the same time, we're activating an adhesive that will adhere that backing to the front and take that sample and basically compartmentalize it into all of those different wells of the Quanti-Tray. That tray will then get incubated. And then after incubation, the tray is removed, results are read, and then looking at the most probable number table for that tray, results can be obtained. So now getting more into the test. So there are, again, 3 different presence-absence quantification tests that I'm going to talk about right now, which is Colilert, Coli18, and Colisure. So starting off with Colilert. Again, Colert's a 24-hour test for detection of total coliforms and E. coli. There are several approvals, both from the EPA and is also found in standard methods, section 9223B. The water matrices that it's approved for include drinking water, groundwater, recreational waters, both fresh and marine, and then finally for wastewater testing for E. coli only. Colilert-18 is very similar, except that it is an 18-hour test. It also can be used for fecal coliform testing if that is required. Again, it is EPA approved and still found in also found in standard methods 9223B. Colilert-18 is also an ISO standard, 9308-2. Again, it's approved for drinking water, groundwater, recreational waters, again, fresh and marine, wastewater for E. coli, and also wastewater for fecal coliform testing approved by the EPA. Now, the way that Colilert and Coliform work are basically, we have this reagent powder that has a bunch of different ingredients in it, kind of grouped into nutrients and other things to help the bacteria recover. So when we're talking about that lag phase, where they are repairing themselves to get ready for cellular division, at that point, like all these nutrients are available in the media to help it get to that through that process. There are also buffers to help keep the pH at an optimal level, and suppressants to prevent other organisms from interfering with the test. And then the next part, which is the part that is most important for us for detection, is the nutrient indicator. And that nutrient indicator is what's going to allow us to say whether or not our target bacteria are present. So Colilert and Coliform have 2 nutrient indicators. The first one is O-nitrophenyl beta-D-galactoperoxide, or ONPG. And basically what this is, you can see there are 2 molecules that are bound together. The O-nitrophenyl is the indicator, and then the beta-D-galactoperoxide is the nutrient. So total coliforms have an enzyme present that is beta-galactosidase, and that beta-galactosidase is able to utilize that OMPG. So when that happens, it will bind to the beta-D-galactoperoxide, which will break that bond, and then that nutrient is released and is now floating free. It hydrolyzes and it changes color. So OMPG, when the nutrient indicator bound together, is colorless. And then when that indicator is released and hydrolyzes, it turns yellow. So for Colilert and Coliform, for coliforms being present, we will be seeing a change from colorless to yellow. The second indicator present is 4-methyl-and-bifferyl beta-D-glucuronide. So it's a very similar concept. We have a nutrient and that's bound to an indicator. And now what we're looking for is the presence of E. coli. So E. coli is a total coliform. So it contains the enzyme in order to utilize the OMPG. And then E. coli has a second enzyme that we're looking for, which is the beta-glucuronidase. And this will allow it to utilize the 4-methyl-and-bifferyl beta-glucuronide, or the MUG. So same thing, that enzyme will be able to bind with that nutrient, and then that bond between the nutrient and the indicator is broken, and that indicator is released and will hydrolyze. Unlike the OMPG, which changes color, MUG is actually a fluorescent test. So when the nutrient and indicator are bound, it does not fluoresce. and then when an indicator is released, it will now fluoresce. It becomes that's that 4-mu molecule. So coliforms and E. coli, which is a coliform, will turn Colilert nicotine yellow by utilizing the OMPG. And then E. coli will also utilize the mug, which will cause it to fluoresce when exposed to a UV light. So for running Colilert and Colilert 18, basically it's incubated at 35 degrees C, plus or minus half a degree C. The different tests, again, have different reads. So Colilert is a 24-hour test with results valid up to 28 hours. Colilert 18 is an 18-hour test with results valid up to 22 hours. When reading results, whether it be presence absence or the Quanti-Tray, the first thing you want to do is look for any yellow color. So yellow, again, we're looking for that OMPG utilization, which will tell us that coliforms are present. If we find that we have vessels that are yellow or we have trays with yellow wells, the next thing to do is be put under a 365 nanometer UV light. And now look to see if those yellow wells are also fluorescing, because yellow wells with fluorescence indicates a positive for E. coli. So again, start off with yellow to look for coliforms. Then if you have yellow, look under a UV light and see if those yellow wells or vessels also fluoresce. For Colilert and Colilert 18, there is a comparator available. So like any test, there can be sometimes kind of variations in appearance of a positive result. So what a comparator does, it allows you to see the minimal amount of yellow and fluorescence in order to call a sample positive. So if you have any question whether or not that well or that vessel has turned positive, you would put it next to the comparator. And if it's equal to or greater than the comparator, then it'd be considered a positive. There is a comparator available for both tray types, the 51 well and the 97 well tray, and also for presence absence vessels. Okay, switching gears a little bit, looking at Colisure. So again, Colisure is used for total coliform and E. coli testing. It has a different time for results. It's valid from 24 to 48 hours, so it's a longer read window. It is also EPA approved and found in section 9223B of standard methods. Approvals are only for drinking water and groundwater. So Colisure acts in a similar manner as Colilert and Colilert 18, where we have a reagent that has all those nutrients and supplements to help the bacteria during that lag phase and log phase. Also has suppressants, and it has the nutrient indicator system. The difference here is that we look for a different indicator for coliforms. So for Colisure, Colisure has chlorophenyl red beta-D galactoperoxide, or CPRG. And it's the same concept. We have that same nutrient that we have in Colilert and Colilert 18, but here the indicator is different. So this indicator, the chlorophenyl red, when it's bound with the nutrient, will appear yellow. Coliforms are present. We have that beta-galactosidase enzyme binding with the nutrient, and that indicator is released. It hydrolyzes, and that chlorophenyl red will turn red magenta. So for Colisure, when we're looking for coliforms, we're looking for that utilization of the CPRG, turning the sample from yellow to red. And then Colisure also contains mug for the detection of E. coli. So that's the same as for Colilert and Colilert 18. So Colisure is also incubated at 35 degrees C, plus or minus half a degree. And again, the big difference for Colisure is the time to results. Colisure has results valid from 24 up to 48 hours of incubation. So reading results, again, we're looking for that red color saying that the CPRG has been used by the coliform. So magenta red indicates a positive total coliform. If you find a vessel that's red, or if you're using Quanti-Tray and you find wells that are red, the next thing to do would be put it under a 365 nanometer UV light. And again, look to see if those magenta colored wells or vessels also fluoresce, indicating a positive for E. coli. So for Colisure, if you want to find red coliforms, red with fluorescence is E. coli. So now talk a little bit more about the quantity trays. So when you have your trays, you have your results, you count them. The next thing to do is go to the associated MPN table, or most probable number table, to find your estimated count for that sample. So when we talk about the 51 well quantity tray, basically the MPN table for this, the first column on the MPN table is the number of positive wells that you're finding in your tray. The next column over is your most probable number for that sample based on the number of wells that are positive. So for example here, if you had your 100 ml sample and you're testing it with Colilert, and you found 9 yellow wells, and then after that you look under the UV light and you find that 5 of those yellow wells also fluoresce. So what would your MPN be for E. coli for the sample? So again, we're looking for wells that are both yellow and fluorescence for E. coli. So if we had 5, we would see that our MPN for the sample would be 5.3. Now, Quanti-Tray 2000 is a little bit more about an eye chart because we have 97 wells total. Basically, the way the tray is divided, at the bottom of the tray, you have 48 small wells, and then the top of the tray has 49 large wells. So when you're counting the Quanti-Tray 2000, you need to count the number of positive wells that you have on both the large and the small. So count your large wells, record that number, then count the small wells, and then record that number. Then you go to the MPN table for the Quanti-Tray 2000. The leftmost column is the number of large positive wells for your tray sample. And then the top row is the number of small positive wells. So basically you find the number of small and the number of large, and then you meet them up, and that will tell you what the MPN is for that sample using the Quanti-Tray 2000. So another example here, you're running Colilert 18, and you find that you have 9 large wells and 6 small wells that are yellow. So what would the MPN be for total coliforms of the sample? So again, you'd go to the 9 large on the first column and then the 6 small on the top row. Come together, and you'll find that the result for this sample is MPN of 16.4. So over the years, one of the questions that we often hear, especially for people who are moving over from a membrane filtration method and moving to an enzymatic method, is that I've been reporting at CFU and now I'm going to be reporting at MPN, and is this going to be a concern because these are different results? But it's not really different at all. What MPN and CFU are basically a label or a unit associated with your result. Basically, what it's going to tell you if you see a result with an MPN is it tells you that that result was obtained using a liquid-based media, such as Colilert. Whereas if you see CFU, which is a colony forming unit, that tells you that that number was obtained using a solid media, such as an agar plate that either has a filter or not. So basically what you're doing is you're just attaching a label to your result, and then that tells people how that result was obtained. They're can both be used. They're both accepted. And the other important part is that they're both an estimation. They both have a confidence range built around them. So neither one is an exact count, because that's not possible, because there's a lot of factors that play into obtaining a result. So it's the best estimation of the result for that sample. We also have an ISO 6107 2021, further strengthening this notion that these are can both be acceptable and used. So as culturable microorganisms per volume can replace CFU per volume or MPN per volume when given a result. And this handout is available in the handouts tab of this presentation. Okay, switching back to quantity trays. So the advantage of the quantity tray quantification system over membrane filtration or even multiple tube is the counting range that is available. So the 51 well quantity tray will allow an MPN result of up to 200 per 100 ml sample. And the Quanti-Tray 2000 gives you a counting range of over 2,400. So having these large value ranges that you can obtain used in the system reduces the need for doing dilutions. Whereas with memory filtration, you might have to do 2 or 3 dilutions per sample in order to get a usable number. Not to say that you still won't have to do dilution if you're using a Quanti-Tray, especially when we're talking about wastewaters. Wastewaters can have very high numbers. So dilutions can be done using the enzymatic methods with the Quanti-Tray system. The important part here is that that dilution is done to the sample before the reagent's added. So in case if you wanted to do a 1 to 10 dilution, you would take your 10 mLs of sample, add that to 90 mLs of your sterile diluent, and then you would add the reagent, and then go through the process as before, dissolving, adding to the tray, sealing, and incubating. And then after you take out your tray, you read results. You just want to make sure that you correct for whatever dilution factor you use for your test. So if you did a 110 dilution, then you take your result and then multiply it by 10 to get back to your value per 100 ml. All right, so switching gears, I'm gonna go quickly over our new product, IDEXX Tecta. So IDEXX Tecta is an automated microbiological testing system. It is a self-contained unit that basically is a incubator and reader all in one. Again, depending on the contamination of the sample, you can have results as quickly as 2 hours and up to 18 hours. Because of its automated system, and also being able to get those results quickly in certain situations, it is ideal for kind of like that non-routine workflows, like when you have water main break, construction retest, and then rush jobs. The other advantage of the system is that since it is an automatic reader, it allows you to have results coming out over the weekend without having to worry about scheduling for staff, or especially right now where there could be staffing issues. It has the ability to do those reads that way you don't have to worry about having somebody come in and doing them. The system can also email results to users, so that way they can have the result without having to actually come in to read from the machine. The Tecta is also approved by US EPA for detecting of coliforms and E. coli in drinking water. So a little bit how it works. This is an enzymatic test, and it works on the similar principle as Colilert does. It is slightly different in that the indicators that we're using are different. We're still looking for the same enzymes that are present in coliforms and E. coli. We're just looking at a different way of visualizing them. So in the case of this, the Tecta test, we have fluorogenic substrate for both the coliform detection and for the E. coli. But again, we're still looking for those same enzymes that we look for using Colilert, Coli-18, and Colisure. The indicator here is a little different. It is hydrophobic. And so what that means is basically it doesn't want to be in water. So once it is released from the nutrient, it will actually collect at the bottom of this cartridge device. And what happens is the machine will then look for fluorescence. And once it sees that, it will trigger it to be a positive and then record that result. What this does, it also eliminates human interpretation. And again, it's able to see results quickly if there is a high amount of contamination. I highly suggest that you visit the link that's on this site, on this page, and look into registering for an upcoming webinar that will go into more detail about IDEXX Tecta. That will be held on December 8th. All right, I'm going to touch quickly on quality control. So when we talk about any test, we want to make sure that when we receive it, it is going to perform as expected. So when we talk about most QC testing, what we want to focus on for media is we want to ensure that it's going to detect the bacteria that we're looking for and that it won't detect other bacteria or other organisms that it's not intended for. So when doing this kind of testing, we would always suggest to use a type strain from a national collection like ATCC. This way you can be assured that the bacteria that you're using is correct. IDEXX also offers a QC line of products that will match up with the different products that we do sell. There is for example, there is a total coliform E. coli kit that will give you a positive for E. coli, a positive for total coliforms with Klebsiella varicola, and then a non-target organism, Pseudomonas aeruginosa, which should show no interaction for results. In addition, we have a very high commitment to quality, and part of that is all of our products will have a plethora of testing done from our quality control lab, and that will be results that will be generated to create a certificate of analysis. So every product that we sell has an associated certificate of analysis that can be obtained from our website. There's a link here on this slide, and you can also just go to idexx.com/water, and there will be several locations where you can look for certificate of analysis. And basically what you want to do is just we'll have a search field. You type in the lot of the product that you're looking to obtain the C of A for, and you hit request certificate, and then it will come up with links that will lead you to a PDF for that C of A for that specific lot. And what that will do is we'll show you the C of A for that lot, which you can then either download and save, or you can print and have for your records. And again, it's going to go through all the different testing that is done internally to ensure that the product is performing as intended, has the correct appearance and pH, and all of that, and whether or not that's coliform free in the case of Colilert, or if it's sterile in the case of Quanti-Tray. So all of those results will be on that certificate. And then you can have that for your records. And that's basically what I have for today.
Awesome. Thank you very much, Shawn. Great presentation. As you know, our customers often have a lot of questions about this topic. So if you don't mind, we can take a look at some of those now.
Yeah.
Okay, great. So before we start, if you're watching this webinar on demand and you have any questions, you can always contact us at water.idexx.com, and we'll be more than happy to answer those for you. And we'll get those addressed for you. Okay, so let's see, Shawn. The first question I have is, earlier you mentioned gram-negative bacteria. Can you just briefly talk about what gram-negative means?
Yeah. So bacteria kind of fall into two groups in very simplistic terms. We have gram-negative bacteria and gram-positive bacteria. And this all came from an individual, Dr. Graham, who came up with a way to do a quick look at differences in bacteria. And one of them is looking at their cellular wall. And basically that's what the difference is, that gram-negative bacteria have a different cell wall than a gram-positive bacteria. And that just helps us group it into different types.
Okay, thank you for that. And here's a good question. How about this? How important is it to kill the chlorine with the sodium thio for presence-absence testing with Colilert? Would it affect the results if vessels containing no sodium thio were used during collection?
Good question. So it's very important. If you have treated water that does have residual chlorine in it, as long as that chlorine is still present in that sample, it will impact the bacteria. So the point of the chlorine in there is to basically it's punching holes and breaking down the bacterial wall cells. So we want to make sure that that's not happening as we're running the test, because we want to make sure the bacteria, again, we're trying to get that log phase to be nice and quick, get the bacteria really healthy, lag phase, get that bacteria really healthy so it enters that log phase. So that sodium thio is very important to neutralize that chlorine, so therefore that's not happening anymore and the bacteria can grow. Now, if you're collecting a sample, like a river sample or something like that, where there's going to be no residual chlorines or anything like that, then not having sodium thio is okay. because you're not trying to neutralize anything, that'll be fine. But it is very important when you're doing presence, absence, or quantification that you neutralize any residual chlorine that could be in that sample.
Okay, great. That makes sense. And let's see, how about this? What is the proper method of mixing the Colilert media in a water sample? Would vigorous shaking potentially damage any bacteria that may already be stressed due to that chlorine residual you mentioned?
Yeah, so that's, yeah, I say over my 20 years, I've seen and heard plenty of different ways that people shake or concerns about shaking. I'll say that, even though the bacteria is injured, how hard we can shake and what we do for shaking is not going to injure bacteria much or significantly. So I would definitely say that you can do a very vigorous, relatively vigorous shake, and it won't impact the bacteria. You can also just do a nice swirl, and that should all be fine. Definitely wouldn't suggest something like a sonicator to help with mixing, because that definitely would actually cause some issues. But just how we would mix, it should be fine.
Okay. And I know this is a common question. What happens if in my sample when it fluoresces, but it doesn't turn color, doesn't change color?
Yeah, so that's a tricky one, and I would definitely suggest to reach out to our technical service for some specifics. That could be about your sample and why that would be happening. But in general, there can be other organisms that just naturally create fluorescence that might be present, that could be causing that reaction to occur. But the important thing is if you see a vessel or a tray that you don't see any color, but you do see fluorescence, then that would still be considered a negative for E. coli. It's really important to remember that you need to have both color and fluorescence in order to call a sample positive for E. coli. So if you're seeing no color with fluorescence, then that would just be a negative. And again, if there's any further questions or concerns, I would definitely reach out to our technical service.
Okay, that makes sense. Thank you. And when counting, do you count the large overflow rectangle well above all the large wells? Does that one count as a well?
Yes, it does. So when we talk about like the Quanti-Tray, I mentioned it was 51 well.
Mm-hmm.
So if you look at the tray, there's one size of well that is there's 50 of those total. So the 51st well is actually that large well at the top, that rectangle well at the top. And the same thing with the Quanti-Tray 2000. There's the 48 small wells, they're all the same size at the bottom. And then at the top, there's the 49 large. Again, 48 of those are going to be the one size. And then that large well at the top is actually the 49th well. So you definitely want to make sure either tray you're using that you're counting that large rectangle well at the top as part of your test.
Okay, thanks for clarifying that. How about a question for marine samples? If you have a marine sample, do you have to dilute it to get an accurate reading when you're using Colilert 18?
Yeah, when using Colilert 18, and it's called out in the insert, there needs to be a 1 to 10 dilution done in order to run a marine sample.
Okay. And foaming. There's a question, when using Colisure, do you have any suggestions for reducing foaming?
Yep. Yeah, there's actually a couple things you can do because Colisure is a very foamy media. Going back to that shaking, if you vigorously shake Colisure, it's going to create a lot of foam, and it takes a little while for it to disperse and go away. So definitely with Colisure, if you're going to mix it, I would suggest doing like a gentle swirling kind of mixture, and that will help reduce the amount of foam. In addition, there is an anti-foam that we do sell, and basically you add some of the anti-foam to your Colisure mixed sample, and it will reduce the amount of bubbles. It won't eliminate the bubbles completely, but it reduces it quite a bit, something that's a little bit more manageable. So with a combination of maybe some gentle swirling and anti-foam, that should take care of any kind of foaming issues that are happening with the Colisure.
Okay, great suggestions. And one final question for you, Shawn.
Okay.
When reading Colilert 18 early, because it is yellow, can I also read for E. coli, or do you have to wait until the 18-hour time mark before saying the sample's negative?
Good question. So one thing I didn't kind of go over is that so the read window of all of the products, is kind of like, it's a definitive answer. You're good to go. If you were to look at a sample before the beginning of the read window and you find that it's positive, then that is considered a confirmed positive and it's good. In the case of Colilert 18, if you're looking at it and you find that it is yellow, so you know it's coliform positive, you can look under a UV light and see if it also fluoresces. If you see fluorescence before 18 hours with the color, then you have a positive for E. coli, and then you can call that good. However, if it's yellow and non-fluorescent before 18 hours, you have to incubate it, put it back in the incubator, and have to wait up until at least the 18 hours, and then look for the fluorescence again. Because then only then, if you see no fluorescence with yellow at 18 hours, can you say it's E. coli negative. So yes, you can look at it before 18 hours, but if it's not there, you need to go to at least the 18 hours to verify whether or not E. coli is present or not.
Okay, great. Thank you for confirming that, and thank you for answering all your questions.
Okay.
Shawn, great job. I really appreciate you taking the time to share your expertise today. Great presentation as always. And for everyone here watching the recording, if you have not visited the IDEXX Water Academy, we encourage you to check it out. It has a lot of courses, videos, and webinars designed for people in the water testing industry. So again, if you would like to learn more about this topic, it's a great resource for you. And you can visit the Water Academy at idexwateracademy.com. And if you'd like more information about any of our educational offerings, please go to the online education hub, which is idexx.com/water/education. You can see that on the screen. And at this site, you can see all the featured courses that we have at the Water Academy. Also, you can see new and featured articles from IDEXX Currents. And also, too, you can see upcoming webinars like the IDEXX Tecta one that Shawn talked about. And you can register for those at that site. So it's a great kind of one-stop shop to bookmark. And so this webinar, or excuse me, the IDEXX Tecta webinar on December 8th will be our last webinar for 2022. But we will be back in 2023 with many more webinars. So please keep an eye out for new and upcoming sessions. And Shawn, thank you again. And thank you to all of you viewing this. We appreciate it, and take care.